So in this video, we are starting the nucleic acid chapter. Nucleic acid is the fourth of the four major macromolecules of biochemistry. We already did carbohydrates, we did lipids, we did proteins, and now we're doing nucleic acids. And the two nucleic acids we're going to focus on are DNA and RNA. Now, nucleic acids are just polymers of nucleotides. So let's find out what makes up a nucleotide and what is the difference between DNA and RNA, what their functions are, and we'll get into stuff that you may have heard of in biology classes, replication and transcription and translation. But first things first, our what you need to know list says, know how to draw the structure of the heterocyclic amine bases. So DNA and RNA are made up of things called nucleotides. A nucleotide, so remember how we said that proteins are polymers of amino acids and polysaccharides are polymers of monosaccharides, specifically glucose. Well, DNA and RNA are polymers of nucleotides. Nucleotides are made up of three components: a cyclical amine base, cyclical amine base, or we can call it a heterogeneous amine base, and a sugar. It's a pentose sugar, so either deoxyribose or ribose, and a phosphate group. So that's what makes up a nucleotide. The other term we're going to learn is nucleoside. A nucleoside is just these two, a cyclical amine base or a heterogeneous amine base hooked up to, or not heterogeneous, sorry, heterocyclical amine base, meaning that it's got carbons and nitrogens in it. A cyclical amine base and attached to a sugar. That's a nucleoside. The minute you throw in a phosphate, then you've got a nucleotide. Okay, there are a few cyclical amine bases that you need to be aware of. A stands for adenine. G stands for guanine. These are the two purines. Purines are cyclical amine bases that have two rings in them, okay? The other cyclical amine bases are C stands for cytosine, not to be confused with cysteine in the protein chapter. That's an amino acid. Cytosine is a cyclical amine base found in nucleic acids. T stands for thymine, and U stands for uracil. Okay. So these three are the pyrimidines. Pyrimidines are made up of just one ring. These three are one ring, these three are two rings. Okay, so what do they actually look like? All right, here we go. I'm going to start off with adenine. Adenine looks like this. All these structures are in your book. This is what adenine looks like. Notice it's two rings. This is adenine. Okay? Guanine is also a purine. It's two rings. Go. And guanine's got a carbonyl up here, and the amine is down here. So, I can't have a double bond here because that would give this 5 bonds. So we have a hydrogen here. So this is A for adenine. This is G for guanine. All right. Now, the three pyrimidines I'll do those next. Cytosine is a single ring. It has a carbonyl here and an amine coming up over there. And double bond here. This is C for cytosine. Okay? This one over here carbonyl here, carbonyl here, and a hydrogen here. This is thymine. Okay, I don't want you guys to get confused here. This hydrogen belongs to cytosine. There you go. This is thymine. And then finally, the third pyrimidine is uracil. This is what uracil looks like. Looks very similar to thymine, except it doesn't have this methyl group attached. So there's a carbonyl here, hydrogen. Notice it looks, uracil and thymine look exactly alike except for the methyl group. Now, one thing you should know is that adenine and guanine and cytosine, those are found in DNA and in RNA. Okay, they're found in both of those nucleic acids. Thymine is only found in DNA. You never find any thymine in RNA ever. And uracil is only in RNA. Okay, so you should know that. All right, so those are your nitrogen-containing heterocyclic bases, cyclical amine bases. Now, when you hook them up to a sugar, you create a nucleoside. By themselves, they're not a nucleoside. They're just cyclical amine bases. A nucleoside is if you take, so I'm going to just take an adenine. I'll just do one purine and one pyrimidine, but all the purines behave the same. All the pyrimidines behave the same when we're hooking them up to a sugar. So this is adenine, remember, and I'm going to hook it up to a ribose sugar. Now that's another key difference between RNA and DNA. RNA always has a ribose sugar. Remember ribose from the carbohydrate chapter. DNA always has a deoxyribose sugar. Okay, now we have to turn those sugars into rings. So we have to create a hemiacetal just like we did in the carbohydrate chapter. So if I'm going to hook it up, let's say to a ribose, carbon 1 and carbon 4 of this pentose sugar are going to form the hemiacetal. Carbon 5 is out of the ring. This happens to be a beta ribose. This is carbon 1, 2, 3, 4, 5. This is a beta ribose. See this H and OH, like we've seen in everything so far in biochemistry, anytime we're creating some bonds, we're removing a molecule of water, HOH. So we get rid of this OH, get rid of this H, and you have this bond. You've just created your first nucleoside. Now, instead of calling it adenine, because it's hooked up to a sugar and it's now a nucleoside, this is called adenosine. The name changes, adenosine. All right, so now I'm going to do a pyrimidine. I'll just choose thymine. And remember how we said that thymine is only found in DNA? So we have to hook up thymine to a deoxyribose. So let's go ahead and do that. Just so I could show you how these things hook up, how they create their bonds. All right, so thymine's got a carbonyl, a CH3. Okay, there's our thymine. I'm going to do a beta deoxyribose. Remember, deoxyribose at carbon 2 doesn't have an OH. So it's just two H's here, so we don't even show them. Okay, again, the carbohydrate chapter. So this is carbon 1, carbon 2 of deoxyribose, carbon 3, carbon 4, carbon 5. Carbon 4 and carbon 1 form this hemiacetal here. All right, HOH, get rid of them. And now we're hooking up this nitrogen of the thymine to carbon 1 of the sugar. Because this is a deoxyribose, you always have to put a lowercase d in front of the name. And because now we have a nucleoside, because it's hooked up to a sugar, the name thymine changes and it's thymidine. Notice the ending. So adenine becomes adenosine. Guanine becomes guanosine. Uracil becomes uridine. Cytosine becomes cytidine, and thymine becomes thymidine. So names of the cyclical amine bases change when they become nucleosides. Anytime one of these A, G, T, C, or U are hooked up to a sugar, pay attention to what sugar you're hooking it up to. If you have a uracil, you cannot hook it up to a deoxyribose. It has to be hooked up to a ribose. If you have a thymine, it has to be hooked up to a deoxyribose, okay? If you're hooking up any one of the sugars to a deoxyribose, make sure you put a D there. And the only one, sorry, any one of the cyclical amine bases, not sugars, any one of the cyclical amine bases, if you're hooking them up to a deoxyribose sugar, you gotta have a D there. The only one that can't be hooked up, remember, to a deoxyribose is uracil. Other than that, you can have D-adenosine, D-guanosine, D-thymidine, D-cytidine, no problem. With uracil, it's just plain old uridine hooked up to a ribose. All right, so that's the structure of nucleosides. Next video, we'll do nucleotides.